To learn how to calculate allele frequency and start looking at the Hardy-Weinberg equilibrium, let's look at an example population of mice that has two alleles for fur color, CB and CW. CB would be black, CW would be white, and this gene shows incomplete dominance, so CW would be gray. If we look at a sample population of mice and we see that 180 have black fur, 240 have gray fur, and 80 have white fur, we can use this population information to calculate the frequency of the two alleles, CB and CW. We see that this sample population has 500 individuals, but keep in mind each individual has two alleles for each gene, so that means there are a total of 1,000 alleles in the population. Let's start by looking at how to calculate the allele frequency for the CB allele. Remember, there are two genotypes that contain CB alleles. Homozygous CB, which there are 180 individuals with that genotype, times 2 is 360 alleles. And then the CB-CW, the heterozygous genotype, contains 240 individuals, each with one of those alleles. So if you add 240 to the 360, that gives us 600 alleles for CB. Now that we know the number of alleles in the population, we can calculate allele frequency. You divide 600 CB alleles by the total of 1,000 alleles in the population. That gives us 0.6 as the allele frequency for the CB allele. We can do the same thing to calculate the CW allele frequency by first calculating the total number of CW alleles in the population. There were 80 individuals that were homozygous CW, so 80 times 2 is 160. And then there were 240 heterozygous individuals, so that would be 240 CW alleles to add to the 160 alleles. This gives us a total of 400 CW alleles in the population. The final step is to divide 400 CW alleles found in the population by the total number of alleles in the population, 1,000. And this gives us an allele frequency of 0.4 for the CW allele in this mouse population. In a population, the sum of all the allele frequencies for a gene equals 1. So in this case, 0.6 plus 0.4 equals 1, the allele frequencies for the CB allele and the CW allele. When there are two alleles for a gene in a population, generally it's denoted with a P to represent the frequency of a one allele and Q to represent the frequency of another allele. So P plus Q equals 1. Remember, these frequencies represent the prevalence of the alleles in the gene pool, or the total alleles for a gene in the population. Now we can look at how knowing allele frequency can be used to calculate the Hardy-Weinberg equation. So if we use P and Q to represent the two alleles possible in the population, we can use a Punnett squared to represent how the Hardy-Weinberg equation is created. So P times P, or the chance of inheriting those together in the population, is P squared. The chance of inheriting P and Q together is PQ. And the chance of inheriting Q and Q together is Q squared. Again, remember, this is calculating the frequency or the percent of a population that would have these expected genotypes, and not the offspring of a parental cross. The probability of any of these outcomes occurring in the population has to equal 1. So P squared plus 2PQ plus Q squared equals 1. Now if we apply this same information to our example problem from earlier with one allele frequency of 0.6, which would represent P, and the other that is 0.4, which would represent Q, we can calculate P squared, which is 0.6 times 0.6 or 0.36. Then we can calculate PQ, which is 0.4 times 0.6 or 0.24, as well as Q squared, which is 0.4 times 0.4, which equals 0.16. And then we can add all of these probabilities together, 0.36 plus 0.48, remember 0.24 times 2, 2PQ, plus 0.16 equals 1. This is a great way to check your answers and make sure you've calculated your allele frequency correctly. So to summarize, we know that the sum of all of our allele frequencies for a gene in a population has to equal 1, so P plus Q equals 1. We also can see how this Punnett square represents the origin of the Hardy-Weinberg formula, P squared plus 2PQ plus Q squared equals 1. When you're using formulas to solve frequency problems, remember the outside of the Punnett square represents the allele frequencies in the population, whereas the inside of the Punnett square information represents the percent of a population with a specific genotype. So if we look at an example, if in a mouse population 64 out of 100 mice have white fur, so CW, we can calculate the percent of the population that have that genotype. So 64 divided by 100 is 0.64. That's our Q squared value. So we can find the square root of that to identify Q, which is going to be 0.8. And then, because we know P plus Q equals 1, if we do 1 minus 0.8, that can tell us our P is 0.2. From there, we could calculate the percent of the population that would be expected to be homozygous, or P squared, or be heterozygous, PQ. Then we can apply the Hardy-Weinberg equation to check our answers to make sure everything equals 1. Knowing this information allows scientists to identify if the frequencies of alleles in a population are remaining constant from generation to generation, meaning they're in Hardy-Weinberg equilibrium, or the population's allele frequency is changing from generation to generation, showing that the population is evolving.